Excellent. So I'd like to get the important bit out of the way first in case I run out of time at the end. I am currently in the process of completing my PhD. I'm looking for a postdoc. In 4 weeks' time, I'll be leaving the lab. You might say I'll be leaving like a bat out of hell, and I'll be gone with 4 minutes. So my project, we've been using yeast, budding yeast, Saccharomyces cerevisiae, and human embryonic kidney cells to characterise roles for EF1A in controlling eukaryotic cell growth. So I'm sure you're all aware of what EF1A is, but just to remind some of you may have forgotten, it's 52 kilodalton G protein, and its canonical role is, as the name suggests, in translation elongation. Up here you can see it in its active GTP bound form, and it delivers the amino acylated tRNA to the A site of the ribosome. But this isn't actually a one-trick pony. It's known to be involved in an array of other cellular functions, including actin bundling. So, as I said, we're using two systems. We're using yeast and we're using mammalians, the human system. To complicate our story, EF1A exists in two isoforms in mammalians. So we have EF1A, and this is universally expressed throughout the body. EF1A1, and we have EF1A2, which shares 92% identity with 1A1, and it's only expressed in skeletal and cardiac muscle and in neurons of healthy individuals. But as shown here, EF1A2 is also found at elevated levels in a variety of cancers. So it's found in 60% of breast tumours and 40% of ovarian tumours. And Jonathan Lee's lab in Ontario, in Canada, they isolated this oncogenic isoform, and when they injected this into nude mice, they showed that EF1A2 overexpression alone was sufficient for induction of tumorigenesis. So here, I'd like to highlight that in S. cerevisiae there are two genes that encode an identical protein, so it makes our story a bit simpler in yeast. So what happens when we overexpress this in yeast? So we've overexpressed the yeast isoform in yeast, and what you can see here is in a logarithmically growing population, we have a nice 50-50 split of diploid and haploid cells as expected. And when we overexpress EF1A, we get this increase of diploid cells, but also we get this third population here, which have had their undergone chromosome duplication, or whole genome duplication again, but for some reason the genome hasn't actually been segregated. And here's another nice result. So I overexpressed the human oncogenic isoform in the human embryonic kidney cells, and this was actually quite a late night result that I was trying to get before another talk. And so when I actually got this result that showed conservation of an increase in genomic content was observed when we overexpressed EF1A2, you might say that I also suffered from a bit of a night fever. But that was me again. So what is this actually doing in the cell? So in a happy wild type cell, this is a highly simplified schematic, so bear with me if I've missed out some components or whatever. But the cell is able to sense its nutrients in the environment, and this is signalled by the TOR pathway, and this promotes cell growth, and the cell cycle progresses quite happily. When we overexpress EF1A in yeast, we have data that suggests that it's potentially interacting with the dynactic complex, with the ego complex, and with various other components in the TOR pathway. And so what we're seeing is a proportion of the population that is arrested at what we think is G0 in the cell cycle. So here's our data, lovely Singer data that we generated in the Singer robot when we had it on trial. And so this is the data that implicates the dynactic complex. And so what we did was we overexpressed the EF1A in an array of strains that have been deleted for genes that have been known to control or interact with actin. So up here you can see our wild type, and when we overexpress EF1A, we get a reduction in growth rate. And then here, I'd just like to highlight ARC1. So this is the backbone complex of the dynactin complex. And what we see is when we delete ARC1, we actually get a rescue of the growth. So we're showing a synthetic interaction between ARC1 and EF1A here. Moving on, we've also got some data, or present some data here, that shows an interaction with the ego complex. So this is involved in amino acid sensing. And similarly to the ARC1 dynactin from the dynactin complex, what we see here is we see, this is relative to the wild type growth rate, we see that when we overexpress EF1A, we get an increase of approximately 25% in the doubling time. When we delete ego1, we see an increase of approximately, 20%. And then when we overexpress EF1A in this ego1 deletion, we get a recovery right back down to wild type. So this suggests another very strong interaction with this complex. So to summarise, we're able to utilise SRViziate to model the interaction between EF1A and other cellular components. We've identified two highly conserved complexes as potential interactive partners of EF1A in yeast, and that's the dynactin complex and the ego complex. If you've got any further questions, I've got a poster in the third room on the right, which shows some of our metabolome and transcriptome data. And that just leaves me to thank my supervisors, Campbell and Tobias, everyone at KFG, BBSRC for funding my PhD, you guys for funding this session, and also for Medium, who kindly sponsor our website. Any questions?